Hello and welcome to Life Science Prep. Today we are going to learn about tetrad analysis, which is a very important topic from Unit 8 Inheritance Biology. If you have seen previous year question papers, there's one or two questions from this unit. So it's a very important topic and you should not skip it. So under tetrad analysis, we'll be learning about ordered and unordered tetrads. In this video, first we will learn the concept of ordered tetrads and then solve some problems which have appeared in previous year question papers. So now what is a tetrad? We already know about fungal cells being haploid and they can reproduce through asexual as well as sexual reproduction. So here we have got two haploid fungal spores which will fuse together to give us a diploid zygote. This will further replicate to give us chromosomes. And these chromosomes will then undergo meiosis to give us 4 haploid spores. That is why we call it a tetrad 4. This is enclosed in a sac called as an ascus and this is seen especially in ascomycete fungi. This tetrad may further undergo mitosis and give us 8 haploid spores and this is called as octad. Now why is this important? Because by studying this ascus and the spores we can get maps, which is a map is basically the position of a gene on the chromosome. And second thing, we can know whether two genes are linked or they are independently assorted. So the linkage can be studied. How is this done? Researchers dissect the asci and they study the spores. Now we can classify the fungal spores as unordered or ordered tetrad or octads. Now what is an unordered tetrad? Here you can see we have an ascus sac having 4 haploid spores. Now these are arranged randomly because there is a lot of space in the ascus. Okay, so the ascus which provides enough space for the tetrads or octads to randomly mix are called as unordered tetrads. This happens in Saccharomyces cerevisiae, Aspergillus nidulans and some algae like Chlamydomonas. Now what is an ordered tetrad? In this case, the ascus is very narrow and does not have much space. So these spores, they cannot mix. They are ordered as and when they are formed. So these two are very related and these two are very related. So a very tight ascus that prevents spores from randomly moving around is an ordered tetrad and it is seen in Neurospora crassa. In this video, we will be learning about these ordered tetrads. Why is it important? Because it helps us to map the distance between a gene and a centromere. So basically, you know what's the centromere? If you have a chromosome, the center part is called as the centromere. If we have a gene anywhere on the arm of the chromosome, we can find out what is the distance from the centromere using this ordered tetrad analysis. So let us take an example. Here we have got 2 haploid spores with different alleles for the same gene. So here we have a capital A and here we have a small a. After undergoing replication, it will duplicate and you will get these 2 copies of capital A and 2 copies of small a. Then when it undergoes further meiosis 1, there will be a cell wall which is formed around these chromosomes. After meiosis 2, they will separate out and you will get 4 haploid spores. If it undergoes mitosis, you will get 8 spores. So this will be our tetrad and this will be the octad. Now if you look at this, it has 4 capital A's and 4 small a's. So we have a ratio of 4:4. So this 4:4 arrangement is seen in first division segregation. So these ascus are called as FDS cells or an M1 pattern. So this 4:4 ratio is very important to solve different questions. So you cannot forget this. And you have to link 4:4 arrangement to the FDS. Now let us look at another case, that is second divisional segregation, which always has crossing over. We'll take the same example. We have got 2 haploid spores. One is having the allele capital A, the other one is having small a. So after replication, you will get it duplicated in this manner. We have capital A, small a, small a. Now since we have to have crossing over occurring here during meiosis, we will do crossing over between these two nearest arms. So if there is crossing over, this arm will remain the same after your meiosis and it will remain as capital A. But in this case, there will be crossing over here downwards and you will get here small e. In this case, again, there's going to be crossing over and this is going to turn into capital A and this one is going to remain the same as it is small e. After your mitosis, this is going to duplicate and your first capital A is going to give you 2 copies. So you'll have 2 copies of capital A, 2 copies of the small a, again 2 copies of capital A and 2 copies of small a. Now let us look at this ratio. It is 2 is to 2 is to 2 is to 2. Okay, so for second divisional segregation, we have the ratio 2 is to 2 is to 2 is to 2. This was in the case if there was crossing over between the nearest arms. Now let us take another example wherein there is crossing over between two different arms. Here we have a haploid chromosome, another one with 2 different genes. After replication, it will duplicate. Now let us take the crossing over between this arm and this arm. We see this as capital A and this one is small a, small a. After your meiosis, this one will remain the same, so we'll get capital A. In the second case, it's going to cross over and take this part of the arm, so it's going to be small a. This one is going to remain the same, there's no crossing over, so it's going to be small a. And in this case, there is crossing over all the way here, so you have got capital A. Now, after mitosis, this will duplicate. So the first arm, that is capital A, will give you 2 copies of capital A, 2 copies of small a, again 2 copies of small a, and 2 copies of capital A. So now in this case, the ratio became 2 is to 4 is to 2. So in the case of SDS, we can have 2 types of ratios, 2 is to 2 is to 2 or 2 is to 4 is to 2. So both of these are related to the second division segregation or the M2 pattern. Now second division segregation is very important to us because it gives us the percentage of SDS ASCII can be related to the map distance. So we can get a formula. We can map the distance of the gene from the centromere by multiplying half into the number of SDS ASCII which are there upon the total number of ASCII into 100. So now let us understand this looking at some problems which have appeared in the paper. So this question came recently in 2024, that is this year. It says the figure summarizes the observation following a cross between 2 haploid strains of Neurospora crassa having alleles capital A and small a respectively. So they have given us a diagram which is very easy because they have explained everything in the diagram. They have given us statements which we have to tell whether they are correct or not. Combination of the statements are: in 1, segregation of alleles occurred in anaphase 2. So in this category 1, they are saying segregation of alleles occurred in anaphase 2. We will know this if we find this as the SDS sac. So let us first name these according to the spores that we have. Look at this first one. It has got 105 and this is 135. Usually the ones having the most number are the FDS type. Other than having the most number, we can also see the ratio over here. It is 4 is to 4. So we have learned that 4 is to 4 is FDS. This second one, class 5, also is having 4 is to 4 ratio. So it is FDS. We'll look at these. It is 2 is to 2 is to 2 is to 2. So we can say it is SDS. This one is 2 is to 4 is to 2, again SDS. This is 2 is to 4 is to 2, again SDS, and this is also SDS. So what is our first statement? In 1, segregation of allele occurs in anaphase 2. This SDS, yes, this option is correct. If you look at the options given to us, only one has got A. The rest do not have. So here we know this is the correct one, but we should read ahead. Statement B says crossing over between the centromere and the gene occurred in 20% of the myocytes. So they say crossing over has occurred in 20%. Now let us look at the crossing over. Crossing over happens in SDS. So what is the total number of SDS? 16 plus 14 plus 18 plus 12. So we need to get a percentage. So it will be number of SDS upon total number into 100. So this total will come up to 60. We will divide it by the total which is given to us 300 upon 300 into 100 and the answer will be 20%. So this also option is correct. So option A is correct. Statement B also is correct. So that is why we can choose option 1. We'll still read statement C because it could be useful for you in the next exams. With reference to the two non-recombinant parental chromosomes, there are 6 different ways by which they can orient themselves at the equatorial plate. So now here they are saying there are 6 different ways. The different ways to find out is we have the formula 2 raised to n, where n is the number of chromosomes. Here we can say n is equal to 1 because we are studying one chromosome in this question, not much data is given to us. So this would lead to 2. So 2 different ways in which the chromosome can be aligned. So this option is wrong. Statement D says the gene is 20 centimorgans. Centimorgan is the unit of map distance in the chromosome away from the centromere. So it says that your gene from the centromere is 20 centimorgans away. Okay, so for this we have the formula which we have studied previously. Map distance is equal to half into SDS ASCII number upon total into 100. So our answer will be half into 60 upon total, that is 300 into 100. So considering this, answer will be 10. So that is why this is also wrong and our correct answer is option 1. Now let us look at another question which came in 2023. A cross was carried out between two strains of Neurospora carrying alleles capital A and small a respectively. The cross led to the following octad patterns. The numbers in the last row indicates the number of octads observed with a given pattern. So basically they have given us your entire chart of the different alleles which were there and the total number. So these are total 8 which we have to check the ratio first. You can see here capital A, small a. So what is our question? In what percentage of myocytes did segregation occur at anaphase 2? So we have to find out in what percentage of these we are getting SDS pattern. So again we are going to find out which is the SDS in this. So here in this case our ratio is 4:4. So this became FDS. Again here we have ratio 4:4. This also became FDS. This is 2:2:2:2 SDS. 2:2:2:2 SDS. Here we have 2:2:2:2. Again we have SDS. And here 2:2:2:2 SDS. So we have got all these as SDS which will have to total up 14 plus 12 plus 16 plus 18. So again, we'll use the same formula upon the total. So if you see these numbers, they are very similar to the previous one. Actually, it's the same. So in 2024 and 2023, they're having the same numbers. But only thing what is different is the total here. So we have got total 400 into 100. So here our answer will be again 60 upon 400 into 100. So our answer will come up to 15. So your correct answer is option 2. So you see again a very easy question. Now we'll look at the question which came in 2022. This is in Drosophila, which is little different from what we have studied, but the method to solve it is the same. So in Drosophila melanogaster, traits A, B and C result from recessive alleles that are located on one of the autosomes. The resulting F1 females were test crossed and the following progeny were obtained. Okay, so this sounds confusing, so we'll directly move to the chart, which is quite simple if you look at it. We have A plus B plus C plus. This is one of the parent which they have told you, and it is having 350 progeny. So what is our question here? The following conclusions were made from the above data. Statement A, the order of genes is A, B, C and the distance between A and B is 8.5 centimorgon. The order of genes is A, C, B and the distance between A and C is 8.5 centimorgon. Okay, so basically all the statements first talk about the order and then the distance. So what we have to do is find out the order of these genes. They have written A, B, C, but the order could be PAC, BCA, any of these. So for this, first we have to find out which is the FDS. FDS are the ones which are occurring the most. We have already spoken about this many times. So let's just write FDS where we can see maximum progeny. This one and this one is our FDS. Now, we also have double crossing over about which we will learn in the next video. Okay, so this is one crossover which happens in your SDS. You can also have double crossing over. If you have double crossing over, we'll take the example A plus B plus C plus, and this is the recessive alleles. So we can have double crossover between this and here, two crosses. So after rearrangement, a new progeny will be A plus, then moving down to B, and from B it will go back up to C plus. And the next one will be A, it will be crossed here to B plus and come back down to C. So in this case, you can see that since B was in the center, B changed. The rest are the same. So what we can do is we can check for the double crossing over based on the allele which changes. And the double crossovers are always very rare. So the ones which are having the least, okay, this one 10 is having least. So we can say this is the double crossover progeny and this is the double crossover outcome. Let's look at this A plus B plus C. So now A plus is B and same, B plus is same, C is different here. Let's look at the other one. A, B and C plus. Okay, C plus has changed again. So from this we know that C is in the center. So our series will either be BCA or ACB. Let's look at the options. Statement A says order is ABC. Wrong. Statement B says order is ACB. Yes, correct. Statement C says order of the genes is BCA. This is also correct. The order of the genes is CBA. No, this one is wrong. So now depending on the distance, we can get an answer. But let's look at the options which are given to us. The options say either A, which is not the correct answer, A and D. A is not the answer. Third option says B and C. So actually we have already solved the problem. B and C is the correct answer. So what is to be done with the centimorgon? It is only given to you so that you waste some time. But we will still solve it now so that you know how to solve this problem. If you have to find out the distance between two genes, which we'll be talking about in the next video, we have a formula for that. So for that, we have to know the order, which is BCA. So let's take the FDS, B plus C plus A plus, and BCA. Let's make a cross between the genes that we want to map. So now in this case, I want to map A and C. So I'll make a cross between A and C. So what will be my outcome? I'll get B plus, C plus, and it will go down to A. So B plus, C plus, and A. This case, I will have B, C, and A plus. Now I need to look at the number of progeny which are having this alleles. B plus, C plus, and A. So where is that? B plus, C plus, and A. So it is 35. The next one, B, C, and A plus. I have B, C, and A plus 25. So these are the number of crossovers which are happening between number of progenies having the crossover between A and C. So for my formula, I need to add the number of SDS having that crossover. So it will be 25 plus 35 plus also these double crossovers have got crossing between A and C, which we spoke about here earlier. So we need to add them as well. Plus 10 plus 15 upon the total number of progeny, which is 1000, into 100. Okay, so this is our formula to find out the map distance, total number of recombinants upon the total number of progeny into 100. Okay, so these are all the recombinants that we have. And the answer will come up to 8.5 centimorgon. You can check for the next one between B and C whether it is 21 centimorgon using the same formula. We'll move to the next problem. This is the problem which came in 2019. It is quite simple. I want you to solve this problem and write the answer in the comment section. We'll learn about unordered tetrads in the next video. So till then, like and subscribe.